Okay, good. Hi, everyone. So I just finished giving my Bio 275 lecture. For those of you who took Bio 275 with me, maybe you remember these pool noodles, right? Doing meiosis and crossing over and stuff like that. Right? So let me get these out of the picture so that's not a distraction. Oh, I pushed them off my desk. All right, that's okay. All right, so let's continue talking about the role of the immune system in fighting cancer. I have my chat box open because I was having some difficulty with Zoom earlier today. So if the slides are not moving along properly, just send me a chat message to let me know and I can restart it. Right? So this is an exciting topic because we've talked about how the immune system is our line of defense against transforming cells that no longer have the ability to be able to, on their own, undergo apoptosis, right? And so, as I said last time, if you have a situation where you actually end up over many years with transforming cells that have made it to the point where you have a large tumor, right, in the body, then you know that implies that along the way, somehow mutations that were stumbled upon by these transforming cells enabled them to avoid or evade immune detection. So if that's the case, right, then you need to have medical intervention, because if allowed to continue on, as we've learned, even though it might have taken a long time, many generations of cell replications for a tumor to reach a certain size, right? It's going to reach a certain size. Now it's beginning to really feel the pressure, the limits of being confined in its particular tumor primary in situ environment, both because of lack of vasculature, as well as being contained physically by things like the extracellular matrix and the basement membrane. Once those selective pressures have rapidly selected for mutations that allow for angiogenesis to happen, the wound healing response to happen, and also epithelial mesenchymal transition to happen, right? Then you're going to have really rapid invasion of surrounding tissues and metastases, right? So at that point, medical intervention is necessary. So you can take different types of strong chemotherapeutic agents to be able to, limit things like cell proliferation, right? But many of those chemotherapies, those drugs, are very sort of systemic and global. In other words, many of those drugs, they cannot tell the difference between a cancer cell that needs to be killed versus a healthy cell, right? Which is why lots of times, right, these chemotherapeutic drugs are themselves highly toxic. So in recent years, now when I say recent years, I mean, the past decade, 2 decades or so, there has been more emphasis on actually using the immune system to fight cancer. Right? So the person's immune system, for some reason, has overlooked the cancer up until this point, but that does not mean that their immune system itself is somehow bad or compromised or no longer working, right? So if you can somehow stimulate the immune system to really pay attention to something that it had been ignoring before, meaning the cancer cells, then you might have a very strong weapon against the cancer. Right. So let's look at some key concepts here just to review. Right. So our first line of defense against cancer is the transforming cell itself, right? We expect that transforming cells in our body, they do the right thing and undergo apoptosis, meaning intrinsic signals, internal signals that are coming from within the cell itself in the form of tumor suppressor proteins like p53, halting the cell cycle, allowing for attempts at DNA repair to happen. But if that fails, then p53, right, pushes the self-destruct button and the cell undergoes apoptosis. I've taught you that the scary thing about mutations is that mutations are not just things that happen every once in a while, once in a blue moon, right? Every single day, your body is incurring hundreds upon hundreds of thousands of mutations in the cells of your body, right? Because you have something like 30 trillion cells. Each one is subject to mutations, right? And so just thinking about how many cells every day do the right thing and undergo intrinsically stimulated apoptosis, right? It's kind of phenomenal, but it makes sense because you have to rely upon the cells to take care of themselves like that, right? But if some of the mutations that these cells have incurred end up doing things like disrupting their ability to follow apoptotic signals that are coming from within them. For example, if they have mutations that are hitting tumor suppressor genes, right, and that nuclear option button is not being pressed, then our second line of defense, right, is our immune system. So if tumor suppressor genes like P53 are lost, it is up to our immune system to detect the cancer cell and encourage it, right, through extrinsic messages coming from another cell, encouraging it to undergo apoptosis. So this is like the actions of natural killer cells of our immune system or cytotoxic T cells triggering the caspase enzyme cascades in a target cell, telling it to undergo apoptosis. Other things that I wanted you to keep in mind as we discuss today's lecture is I've taught you that cells are constantly presenting antigens on their surface, right, as a way of advertising to the immune system that either, hey, everything's normal because all the little signals like small peptides taken from recycled proteins, inside of the cell are normal everyday peptides that your body and your immune system has grown accustomed to expecting, and therefore a healthy cell that presents normal signals is left alone, right? But if a cell is transforming and you might have weird mutant proteins, things like that, then you might have funny peptides, right, that are different than what's normally expected. And so your immune system is sensitive to that and can maybe then tell that cell, no, there's something wrong with you, right? We said that antigen expression levels, right, in the cell is what we call immunogenicity, right? And that has to do with detectability of this cell by your immune system, right? The more immunogenic a particular signal is, the stronger your immune system can detect it. Right? We've also learned some examples already that mutations can help cancer cells evade the immune system by doing things like downregulation of the MHC proteins on their surface that are used to present antigens, right? So by no longer presenting or advertising what's happening inside of them, right? That's one way that cancer cells can hide from the immune system. They can also, through other mutations, turn on the expression of certain proteins that maybe they shouldn't normally be making. Things like high levels of TGF-beta or expression of FASL, ligand that end up killing surrounding cells, like the lymphocytes that are coming to try to attack them. We also saw examples where mutations can lead to cancer cells resisting the activity of complement proteins, these proteins in our immune system that normally bind to targets and label them for things like macrophages to come and eat them up. Another one that we mentioned, but we'll look at today in more detail, is recruitment of Treg cells. All right. So in each of these cases, what are we dealing with? Right? We are dealing with a cancer cell that in some way is able to disguise itself so that it avoids detection. Right? So it's very much the wolf in sheep's clothing. I like this painting. It's really creepy, but I like it. Okay, anyhow. So let's talk about this recruitment of Treg cells. So Treg cells are a particular type of helper T cell. These helper T cells are recruited, they bind to a target, and what they actually do is they discourage other types of T cells, like cytotoxic T cells, from coming and attacking the target. Right? You might say, why would we have cells like this in our immune system, right? We have cells like this because if it weren't for the fact that we had Tregs, then our cytotoxic T cells all the time would be making random mistakes and attacking different cells in our body, right? Tregs then are able to kind of keep the noise level or the basal activity level of all of our other lymphocytes in our immune system sort of at a manageable level, right? And it's only when signals rise above that level do you then activate cytotoxic T cells, right? You know, one example that I often give is if you're in your car driving around and you're listening to the radio. I think all of you have done this before. You know, you're listening to a radio station, it's nice and clear, the music is really clear, but then you pass through a tunnel, right? Like this happens to me all the time when I'm driving to and from Kailua through the Pali tunnels. And you begin to lose reception, right? But the interesting thing that happens, you notice, is because it's not just the music gets quieter, right? But it begins to break up and you get a lot of sort of noise, like hiss, right? What is that hiss? That hiss is actually just noise that's always present in the signal, right? All these radio waves going through the air, all the electronics in your car stereo that are processing this, there's going to be just noise, and that's the hiss level. Normally, right, your stereo in your car is calibrated to only sort of pay attention to signals that are beyond that sort of background noise level, right? And so they only amplify then the good music. The problem is once you go through a tunnel, the good signal levels drop and your stereo tries to compensate by raising up, right, its sensitivity. And that's why you then hear the noise level come out, right? And then once you leave the tunnel, right, the strong music signal comes back and your stereo then readjusts. So it suppresses the noise. So that noise is always present in our immune system as well. And our Tregs are the cells that keep that noise level suppressed. All right. So here we have examples of a tumor cell. Right? So these tumor cells have stumbled upon different mutations that result in the expression of certain cytokine messages like CCL22. CCL22 attracts these Treg cells to the tumor cell. So you can see these purple dots are being attracted or are very attractive to these Tregs, these purple ones, right? And so these Treg cells, they come and bind the tumor cell. And once they bind the tumor cell, like this one here, they send inhibitory messages to other helper T cells, other cytotoxic T cells. These are our effector cells, right? That normally should be detecting the tumor and raising the alarm that, that the immune system should attack these tumor cells. Right? These Tregs also release, these tumor cells also release TGF beta, which is attractive to the Treg cells, right? But discouraging to the other types of lymphocytes, like the other helper T cells or the cytotoxic T cells. Right? So this is pretty diabolical because this is actually what our tumor cells learning to use immune cells to its advantage, right, to protect itself by recruiting these Tregs. So one of my friends, former colleague of mine, we did our PhD together at USC in the cancer center. His work actually focuses on Tregs, right? And he's trying to find ways of treating patients who show big evidence of these Tregs being attracted to their tumors. He's trying to develop ways of actually discouraging these Tregs from associating with tumor tissues. Because is that really important? Yeah, absolutely. Here's another Kaplan-Meier plot looking at 3 cohorts of patients who were diagnosed at time 0, all right, with a particular cancer. I forget which cancer this is, what this data is, but that's not that important, right? At the time of diagnosis, when the pathologist looked at their tumor biopsies, they stained for the presence of Tregs. Are there Tregs being attracted to this tumor? And you can see in patients who had high levels from day 1, their primary biopsy at the time of diagnosis, of already having lots of Treg cells being attracted there, their prognosis, right, the high concentration Treg patients, is far worse than patients who, at the time of diagnosis, their tumors showed little evidence of Tregs being attracted there. Right? Okay. So the question is, if the immune system itself, right, isn't necessarily, there's nothing wrong with it, right? A cancer patient doesn't necessarily have somehow a compromised immune system. It's just an immune system that has been fooled into ignoring the cancer cells. Then wouldn't it be amazing to be able to reverse that, right? Get the immune system to start paying attention to the cancer. Because compared to all the different types of chemotherapeutic compounds that target cells to stop proliferation, many of those are very toxic, as I mentioned, right? Think about this. Mother Nature already equipped us with probably the strongest set of mechanisms to fight cancer, and that is our immune system, right? So if you can leverage a patient's own immune system to fight against their cancer, right, that is probably the strongest tool you have to successfully beating it. So how can we therapeutically boost the immune system to better target and destroy cancer cells? Remember that a patient's immune system might be perfectly fine, but tumor cells have stumbled upon combinations of mutations that allow them to evade. Right, so that's our wolf in sheep's clothing, right? That we cannot detect in this flock of sheep, right? This wolf maybe, hey, I didn't even realize, wow, this is how clever that I didn't even realize all along that there's another wolf in the back over there, right? Wow, these clever tumor cells. All right. So one way to do it is if we can go ahead and label these cells in a way that the immune system will now pay attention to them. Those of you who have taken immunology, right, what's a great way to specifically target something, right, with signals that the immune system is designed to detect? Right? Antibodies, right? So if you can use antibodies directed against certain cancer antigens that are on the surface of a cancer cell, you can introduce large amounts of these specific antibodies into a cancer patient, and they will hopefully bind to the cancer cells, and then the antibodies will function as part of the immune system to raise the alarm, right? So that other immune cells will now start paying attention, right? So it's like putting little markers on the particular sheep wolf that you want to identify and highlight. Right. So I think many of you are familiar, at least in passing, with the fact that many of the most popular frontline cancer therapies now, and not only just cancer, but other diseases as well, involve monoclonal antibodies, or MOABs. So this is our introduction to the use of monoclonal antibodies as cancer drugs, right? Because again, right, the purpose of using antibodies like this is to focus the patient's own immune system to pay attention to the cancer that it's been ignoring up until now. And it makes use of existing immune cells and pathways, right? So oftentimes when pharmaceutical companies are engineering these antibodies, not surprisingly, it remains true that the best source of producing antibodies is a living immune system, right? So that's why animals are often used as sort of the resource to generate lots of antibodies, and then these antibodies are purified from the animal and then processed and used as a therapeutic agent. So oftentimes, for example, you will need to produce an antibody in a different animal. But keep in mind, if that protein, an antibody is a protein, right? If that's being produced in another animal, you cannot just introduce it directly into the human body, because your immune system will recognize it as being something foreign, certainly not belonging to what the body's used to seeing. And you probably end up just destroying that antibody as a part of the normal immune response. So that means if you grow or you produce a lot of antibody in, let's say, a mouse, right? So for example, this red antibody here is a mouse antibody. You will need to somehow alter it in a way to make it more acceptable for the human immune system to accept it, right? One way of doing it is to what we call humanizing it. That means you actually take the specific peptide regions of the mouse antibody, right? Oh, and I should say, the way that antibodies are produced in other animals is simply a matter of exposing those animals to the particular antigenic signal that you want their immune systems to focus on making antibodies against, right? So for example, if there are certain cancer antigens on the surface of cancer cells in the human body that you want to produce antibodies against, right? You might be able to purify those antigens, like those peptides, for example, right? And expose something like a mouse to those, and the mouse's immune system will produce antibodies against it, right? So once it's done that, the variable parts of the antibody that give it its specificity, those peptide regions, right? You could just take those and engineer a human version of the antibody. So now the beige region is human antibody, but these regions, right, have incorporated the variable parts that were produced by the mouse. So we call that a humanized version of this antibody. A chimeric antibody might be a little bit more handled a little bit more macro, right? Meaning instead of taking specific peptide sequences, you might actually take the entire region of the mouse antibody, like the entire variable domain of the antibody, and just plop it or attach it onto a human antibody, making it chimeric, right? Or, it could be that these antibodies are originally purified from human sources in the first place, right? In which case, I'm not saying that they actually inject people with antigen and they make these, but they can actually determine, right, certain antibodies that people naturally produce against certain antigens, right? They can try to go ahead and take those and in the lab make cells that produce a lot of these human antibodies. Right? So the nomenclature of these monoclonal antibodies is kind of a funny business because you've probably seen this before, right? Like you're watching a commercial for some drug or for some medication on TV. Pharma companies are really funny when it comes to drug commercials, right? Because drug commercials they usually follow the same kind of formula. It's usually some person who's really happy and or something, they have something wrong with them and they're not so happy, but then in the course of the commercial they get happy and then they're enjoying life, right? So they started off maybe staring out of a rainy window, right? And then by the end they're out playing with friends, flying kites or something like that, right? That's just because typical general public, if you were to actually have a 30-second commercial that explained the molecular pathway of how this drug works, I mean, forget it, right? I mean, you guys might like that commercial and be like, wow, that's cool. But the most 99% of the population would be like, whatever, they'd change the channel, right? So anyhow, it's better to show people sad and then flying kites. But anyhow, so drugs oftentimes have a market name, something that's catchy, right? Like Rituxan or Gazyva or Arzerra. These particular examples here, these three, are drugs that are monoclonal antibodies. The actual antibody name are things like rituximab or ibanutuzumab, right? Or afatumumab, right? And so you've probably seen these before, these funny names that are often in little parentheses during the commercial. You'll see the big name, Rituxan, and then you'll see in parentheses, it'll say, rituximab. Right. So this naming is not just haphazard, even though it sounds really weird, right? There actually is a proper set of rules for naming these. So the way it works is there's three parts to each of these monoclonal antibody names. Right? The last part, if you haven't noticed before, but now hopefully you realize, if it ends in mab, that means you're dealing with a monoclonal antibody drug. Right? The middle part, the Z, the ZU, or the U in these three cases refers to whether this is a humanized or chimeric antibody. So let's look at this here. So chimeric antibodies, right, they use this subsystem of having an XI in the middle. All right, so rituximab is a good example of that. Right? Humanized antibodies have a ZU, right? So things like trastuzumab, which I think some of you have encountered in your article summary assignments. Human antibodies just have a U in the middle, like ipilimumab. Right? The first part of the nomenclature, that is more up to, the imagination of whoever wants to name this at the pharma company, right? That's a little bit more up in the air. Okay, but now when you see these names, right, you might be able to play this little game of figuring out, oh, it's a monoclonal antibody and it is chimeric or humanized. Right? Right. So let's look at some examples of these. So here are monoclonal antibodies for the treatment of B cell cancers. So in certain B cells, so B cells are a type of lymphocyte. In many B cell cancers, these B cells show a common feature of overexpressing this particular protein CD20 receptor on their surface. So as a result, the CD20 receptor can serve as a nice antigen target for antibodies to come and bind to, right? Because other cells in your body wouldn't be expressing as much of this CD20, if at all, right? Whereas these cancerous B cells, they oftentimes express a ton of CD20, right? So you take advantage of it. So these three drugs that we looked at on the previous slide, Rituxan, Arzerra, and Gazyva, they are antibodies that detect and bind to CD20. So here is the surface membrane of a B cell. This is a B cell, maybe a lymphoma cell, for example, or leukemia. Here's the CD20 in gray, right? So this is the surface. It's a receptor, so part of it sticks out. Right? So this unique three-dimensional shape provides several different opportunities to have antibodies recognize it. For example, Rituxan, you can see here, right? So this is the antibody. It's an upside down Y in this picture here, right? It's binding to this particular epitope region of the CD20. Notice how Arzerra, right, also binds to CD20, but it binds to this particular epitope surface, right? Because the complex three-dimensional shape of this antigen provides multiple different possible individual little 3D surfaces, epitopes that different antibodies can recognize. The Gazyva recognizes this part of the CD20. Okay? Look at how effective this is. Okay? If you look at a bunch of cells, B cells, and you rank them from having very low numbers of CD20 on their surface, like all the way down to 0 down here, all the way up to, I don't know, 80,000 molecules of CD20 on their surface. And then you expose these cells to things like these compounds, like or these antibodies like rituximab, and then you actually measure how effectively do these cells get destroyed by the fact that antibodies bind to them. You can very clearly see the more CD20 molecules they have on their surface, the more sensitive they are to antibodies detecting them and eliminating, right? Because this is on the y-axis, the relative percentage number of cells that get lysed or destroyed. In addition, you can use this sort of as an adjuvant therapy. So adjuvant therapies are therapies, I think some of you know this already from your article research, where you have not just one treatment happening at the same time, but a combination of different types of treatment. Most of the time, we talk about it as different mechanisms of treatment. For example, radiation therapy and therapeutic drug treatments at the same time, right? But you can also refer to this as combinations of different drugs. So in this case, what they're showing is that if you use a separate drug, a CD59 inhibitor, okay? Remember, CD59 is a protein that's produced by cells of our body to discourage the complement proteins of our immune system being attracted and binding to the target cell. Right? So cancer cells that produce a lot of CD59, they are resistant to complement proteins binding to them, and therefore they are resistant to the complement-mediated mechanism of our immune system destroying the cell. So look, if you combine Rituxan, the monoclonal antibody treatment, with this CD59 inhibitor, right? So now you're inhibiting the function of the CD59. You can see that it's much more effective than either the CD59 inhibitor alone or the Rituxan antibody alone. Right? Okay. So these monoclonal antibodies are directing the immune system to attack the cancer cell, right? So things like rituximab, right? The antibodies will bind to a malignant B cell, And they will do things like, because you have the antibodies now, these are the little brown things bound to the CD20 on their surface, they can recruit things like macrophages or natural killer cells that normally, for whatever reason, have been ignoring the cancer cell, right? And now they will attack the cell and destroy it. So we call this antibody-dependent cell cytotoxicity, or ADCC. Right? So you'll often see ADCC referenced in clinical studies. So now you know ADCC refers to antibody-dependent cell cytotoxicity. All right, and that leads to cell lysis. Also, antibodies can recruit complement proteins, right? So complement proteins then can then result in the lysis of the cell as well, right? As well as the immune system potentially triggering apoptosis, right? Cytotoxic T cells can trigger apoptosis. Right? So I showed you that different antibodies might be able to target the same antigen just using different epitopes, right? So here's another really good example of that. You can combine treatment with different monoclonal antibodies to really go at a particular target antigen at the same time. So we've learned that epidermal growth factor receptors are often overexpressed in tumor cells, right? So that means the EGF receptor might be a good target for certain antibodies. The EGF receptor has many potential target surfaces or epitopes on it, so you can have different antibody drugs all working together. So here you have the HER1, HER2/neu, right? So these are examples of EGF receptors. And here you have cetuximab, right? This is just I guess this is just one arm of the antibody, the light chain and heavy chain of this arm of the antibody. And you can see only this surface here. This is neat, right? Because you can see only this surface here where I'm pointing to with the cursor is where the epitope is, the unique three-dimensional shape that this cetuximab antibody recognizes. Right? Trastuzumab, also known as Herceptin, it actually targets a different part of the receptor, right? It targets down here. Pertuzumab targets over here. Right? So that means a patient, let's say with HER2 positive breast cancer, right? There's no reason why she necessarily has to only get infusion of one type of monoclonal antibody, right? Her treating oncologist might say, I think you're a good candidate. Let's just try going at it with 2 or 3 different monoclonal antibodies at the same time to really try to take care of eliminating these cancer cells. Right? So another way of doing this is of using antibodies is not to necessarily directly bind to the target cancer cell itself, but you can also use antibodies to kind of modulate or control the activation of other immune cells. So for example, T cells like cytotoxic T cells or helper T cells, when they interact with a target cell, they interact because their T cell receptor, so in this cartoon on the left, the gray part is the naive or inactive helper T cell. The yellow part on the bottom is the antigen presenting or APC cell that's going to show it certain things, like, for example, this red dot, which is the antigen that this helper T cell will hopefully recognize and activate against. Right? So the naive T cell needs to see that with its T cell receptor, but at the same time, it needs encouragement to actually activate, just in case this is like a mistake, right? So the antigen presenting cell, in this case, this is a dendritic cell from your immune system, and it will present something like this B7 that is received by CD28 on the naive T cell, right? So it gets a combination of a strong stimulatory signal from the T cell receptor activating against antigen, as well as this encouraging or confirmational signal from this B7 CD28, and those two will activate the T cell. All right. However, on the surface of the T cell, it also has other receptors like this CTLA4, which are inhibitory, right? Because cell signaling is always a balance between positive stimulatory signals and negative inhibitory signals, And it's always a matter of which ones of these competing signals win out that results in a change in the cell's response. Right? So on its surface, it has lots of these CTLA4 receptors as well, these pink ones. When they bind to B7, right, they actually inhibit the cell from activating. Right? So again, you can think of this as another way of the immune system trying to keep noise level down at a manageable level. Right? In order for this T cell to really activate, there really has to be way stronger stimulatory signals to overcome these inhibitory signals. Right? Guess what? Right? One way you can use monoclonal antibodies is, wouldn't it be great if you could somehow prevent these CTLA4 inhibitory receptors, right, from discouraging the helper T cells from activating? That way, your helper T cells would become more sensitive to things like cancer cells, right, and would activate in greater numbers. So how might antibodies designed against these molecules affect T cell responses? Right? You can imagine each one of these molecules, like the CTLA4, right, is a protein itself. That means it has potential epitopes on it that antibodies could recognize if you have the right antibody. Right, so here's an example. So this ipilimumab, right, which is also called Yervoy. I don't know why it's called Yervoy. I like the name, right? But anyhow, right, is an anti-CTLA4 antibody. You can see here in this clinical trial, what they've done. Sorry, this is a mouse experiment, so this is done in the lab. Right? So they injected tumor cells into probably an immunocompromised mouse, right? And then they also start giving it antibody treatments with an antibody that's against CD28 and an antibody that's against CTLA4. Right? And then they follow the average tumor area of this growing tumor. So you see, if they leave the mouse alone, right, it develops its tumors in the red line over here. This is no extra treatment, right? You develop tumors of about 150 square millimeters in area by around, like, day 33 or something like that, right? If you're treating these mice with anti-CD28 antibody, that makes things worse, right? Because let's just go back to the top slide, right? CD28 needs to participate in part of the stimulatory signals for waking up naive T cells, right? So if you have antibodies that bind to CD28 and get between the CD28 and the B7, right, then you lose a whole set of stimulatory signals. Right? And you can see, as a result, the blue line shows that these tumors grow even faster. But if you use an antibody directed against CTLA4, right, let's go back on the previous slide here. Remember, CTLA4, when it binds to B7 from the antigen presenting cell, it actually inhibits the activated T cell, or the T cell, from activating, right? So if you can get antibodies in between the CTLA4 and the B7, right, you can prevent these inhibitory signals from happening. And sure enough, wow, look how good that works, right? Because the tumors in these mice that are being treated with the anti-CTLA4 antibody, they're Yervoy, right? I like that. Right, their tumors don't grow, right? And so you can see out to day 33, they have no tumor growth. So here on the right is a clinical trial in a patient. Right? So here it is pre-treatment. Here you have this large lung mass, which is the tumor. Okay, and following 5 months of treatment with ipilimumab, you can see a dramatic reduction in the mass of the tumor, right? And in this case, all this is an antibody that's helping the patient's own T cells activate, right? Activate stronger than they normally would. Okay. So other types of immune-based cancer therapies include things like bone marrow transplant. So for example, if you have cancer that is hematopoietic in origin, so involving the hematopoietic stem cells of the patient's immune system, giving rise to mutant cancerous, lymphocytes, for example, right? Then you can do a bone marrow transplant. You need to get rid of the patient's sort of defective or cancerous stem cells and then transplant in new stem cells that are wild type, right? That hopefully won't be rejected by the patient's immune system, will actually sort of take root and start replenishing healthy versions of lymphocytes. Right? Bacterial stimulation can also be used. So if you can activate the immune system, kind of give it just sort of like a wake-up call, a good kick in the butt kind of thing, right? Even if it's not specifically telling it to pay attention to the cancer, if you can just get the immune system to kind of freak out and start sort of like, waking up like, what's going on, right? Then maybe it'll somehow notice the cancer when it didn't before, right? So bacterial stimulation can work. So BCG is a strain of bacteria that can be used to kind of wake up the immune system and just hope that the patient's immune system now pays attention to the tumor. And the third one, which is very intriguing and very new, is vaccination. So you might wonder, can you actually vaccinate against cancer in the same way that you vaccinate against, other infectious diseases? Right? So I'm going to talk about two examples of this: Gardasil, which is the vaccine against HPV, and also Provenge, right? Provenge, which isn't necessarily a traditional vaccine, but is a procedure that essentially acts as though you're vaccinating against cancers. So let's start with Gardasil and HPV. So human papillomavirus, or HPV, is the most commonly cause of sexually transmitted diseases. And the majority of infections with HPV, usually resolve in a couple years. All right. In other words, after having viral loads detectable in the blood, after a couple years, usually your immune system has cleared that. However, in longer cases, the viral DNA, as we learned in this class, if it integrates into the genomes of cells that don't actually carry out the viral replication function, then there is the opportunity, right, for mutations to happen and persist as a result of the viral genome messing up the normal cell genome, right? And if that happens, it can lead to things like cervical and other genital cancers, right? So this is what we're talking about here, is that normally infected cells with HPV, right, they will be hijacked and they will carry out the will of the HPV particle and release more HPV, and in the process, these cells will just die, right? But what if the integration of the genome, right, doesn't successfully lead to the cell producing more virus particles, but instead you just have these cells persisting with integrated viral DNA, right? And that can lead to cancer. So because HPV is a virus, you can think about this. If you have a vaccine that protects you against the virus, then that vaccine is not only protecting you against HPV infection, but is essentially protecting you against cervical and other cancers that are caused by HPV infection, right? So Gardasil is a formulation, it's a multivalent formulation of different, basically peptides taken from the different proteins of 9 different strains of HPV, right? So these are the protein fragments that would represent sort of like the most obvious epitopes on the surface of the virus particle that the immune system would normally have a chance to learn from and develop an immune response, right? So if you just take these particular peptides and give that to a person's immune system, then their immune system has the opportunity to learn to recognize these as something that they should build up protection against. But the benefit is you're not giving them the actual, being infected by the HPV, right? You're just giving them the pieces that you want the immune system to learn from. So Gardasil 9 has been demonstrated to be 100% effective in preventing precancerous lesions of cervical tissue, right? So that is astounding, right? How effective this HPV vaccination can be in protecting against the formation of these precancerous lesions. But it's controversial because, just like any other vaccine, in order for it to work, it has to be given before the first time a person is infected with real HPV, right? And that means getting to young people like teenagers, before they are sexually active, right? And so that's why the recommended age now for giving this Gardasil 9 vaccine is like kids who are like 9 or 10, right? And that's controversial because, you know, a lot of people don't like what that implies, right? That somehow makes parents uncomfortable because it somehow implies that they're acknowledging that their 9 or 10-year-old kid pretty soon is going to become sexually active, right? So it's a real shame that there's pushback against this. You know, a lot of parents don't want to have their kids vaccinated with Gardasil. And yet it's being shown to be so effective in preventing the future development of cervical cancers, right? The last example I'm going to end with is this Provenge that I spoke about. So Provenge is another example of how you can sort of vaccinate against cancer. But in this case, we're not talking about, you know, something that comes from a virus or something like that. What we are talking about is certain antigens that are produced by tumor cells are unique to that particular type of tumor, right? We already mentioned that there are these things we call tumor-specific antigens. So in prostate tumors, the carcinomas that form, right, these transformed prostate cancer cells make a particular protein known as prostatic acid phosphatase, or PAP, right? And this is expressed in prostate carcinomas. So here's a histology slide showing carcinoma of the prostate, and they're staining the PAP protein in red, right? So you can see it's very abundant in these prostate cancer cells, and the PAP expression is negligible in other tissues of the body. Hey, so that's a great target then, right? If you can get your immune system to actually target this, that would be really great, right? So what researchers do, or what they've developed, is this technique where they've made a PAP peptide, right, that's sort of fused to this other protein, which is a CSF colony stimulating factor that stimulates macrophages to grow, right? So this is the type of signaling molecule that's very readily taken up by certain cells of our immune system, like things like monocytes, like things like dendritic cells, right? So what they do is the patient goes in and from their blood, right, a lot of blood is taken from them, and from that blood, in the lab, their dendritic cells are purified, right? Monocytes and dendritic cells are purified from their blood. And essentially, in a test tube, these immune cells are force-fed this PAP GMSF ligand. So you can see here, right? So here's the ligand. They call it PA 2024. Right? So it's very readily taken up by monocytes, and importantly, right, it's very readily taken up by dendritic cells that like the blue part, which is the GM-CSF part, but because the red part is attached to it, it just takes it all in there. Right? And it ends up processing these peptides and presenting these peptides on its surface, including the PAP signal. Okay? So then once these immune cells are trained, especially the dendritic cells that have been force-fed this PAP peptide, so now they're presenting tons of this PAP peptide on their surface, those immune cells are transfused back into the patient's body, right? And they go to work. They go to the patient's lymph nodes and the dendritic cells. Their whole job is to show other immune cells, hey, these are signals that we need to pay attention to, right? And now it's almost like through a bullhorn, right? Really amplified, saying this PAP signal is what T cells need to pay attention to. And then the patient's own T cells wake up and go to work and start attacking their prostate tumor, right? The beauty of that, again, is you are leveraging the patient's own immune system to fight their own tumor, which is what Mother Nature designed us to do, right? So those mechanisms of fighting the tumor are natural mechanisms. It's just they needed to be woken up in this way artificially. The other beauty of this is these immune cells came from the patient in the first place, right? So there's very little risk of their immune system rejecting these cells once they are reintroduced in there. So this is very exciting. It's been around now for, I think, 5 years, this Provenge. I like the name of this one too, right? Because it's revenge against your prostate cancer, right? But the downside of this is the cost. Because the way I explained it to you, with these multiple sort of drawing of blood, processing in the lab, and transfusions, right? I think the whole Provenge treatment is over the course of 3 visits, I think over the course of several months. And it costs, I think, about $100,000 to do this, right? And I don't think that, unfortunately, insurance companies, cover it, right? Because it's still considered maybe too new and elective. Right? But this is exciting because as the cost and popularity of these types of treatments expand in the future, I think inevitably we will see these types of immune modulation therapies become cheaper, and more accessible. Right? All right, so the last slide then is just a summary. Right? So these are just examples then of different ways that you can immunize, right, against the formation of tumors, either through the infusion of specific monoclonal antibodies like we've talked about, or actively, right, doing things like being able to put tumor-infiltrating lymphocytes into a person, or like the Provenge, infusing with dendritic cells that are loaded up with tumor-specific oligopeptides to be able to really get the patient's immune system to wake up and pay attention to what you want it to pay attention to. Right? And so that's what I want you to think about, right? The 2 things here. Number 1, I stress again, right, the fact that once you get a large tumor, that means that somewhere along the way, the tumor has learned how to get around the immune system, right? But that doesn't mean that the patient's immune system itself is somehow compromised, right? It's just being fooled. So being able to come up with new therapies that leverage the patient's own immune system to wake up and pay attention, right, is far more powerful than any type of systemic compound or any type of or therapeutic drug that you could give them, right? Because again, you're using what Mother Nature designed us to do in the first place, right? So that's why things like monoclonal antibody drugs are so exciting now, right? And why immune modulation therapies like Provenge are so exciting as well. All right. Okay, let's go ahead and stop there.